Hi viewers, this is Ramesh. Now I am going to speak about the solid waste management technique. I am speaking about the technique of municipal solid waste digestion technique. Lot of techniques available. That is the vermicomposting technique, and then biogasification using anaerobic digestion. Here I am handling about the upflow anaerobic sludge blanket reactor in this study, that is called as a UASB reactor. Everybody knows there is a municipal solid waste generation in all the important cities of India, specifically in Tamil Nadu. Erode is the fast developing city. Huge amount of solid waste generated, around 1200 tons of solid waste is generated in Erode. It is segregated into non-biodegradable waste and biodegradable waste. Around 10 sampling stations are taken for this study. So in the 10 sampling stations, separated waste is separated into biodegradable and non-biodegradable. That non-biodegradable waste are discarded and it is taken for some other study, and biodegradable waste are taken for the anaerobic digestion. The anaerobic digestion is the important one because we are generating the biogas. The biogas is used to rotate the various machines and we are getting the thermal energy. The thermal energy is going to be used for burning. Here I am taking the upflow anaerobic sludge blanket reactor. In this figure you are seeing that dumping yard. Dumping yard is located in Erode town. This machine is segregating the various kind of waste like plastic waste, rubber, glass and biodegradable waste. We are taking the study for only biodegradable waste. Here various techniques are going in the dumping yard. The one of the technique is a vermicomposting. Here you are seeing that after the processing is over, that municipal solid waste is converted into that one of the manure. It is called as a vermicompost. Huge amount of worms are introduced and finally after 48 days we are getting this kind of manure, and then this manure is taken to the various places for the cultivation. Next one, that main topic, that is called as a biogasification. The biogasification basically classified into two important stages. First one is acidification stage, second one is methanation stage. In the acidification stage, there is 6 weeks is needed for the acidification. So the first week and second week, the biodegradable waste are going to be produced some amount of leachate. This leachate is finally diluted and it is going to be converted into the acetic acid. Finally the acetic acid is taken to the UASB reactor. So the 6 reactors are used for the acidification stages, and then finally that wastage is going to be taken for the upflow anaerobic sludge blanket reactor. So the upflow anaerobic sludge blanket reactor is going to be easily convert that acidic stage into the methanation stage. So a huge amount of municipalities nowadays produce the biogasification. In Namakkal, they are constructing that this kind of biogasification reactor. In Palipalayam, this kind of reactor is successfully taking the biogasification from the organic fraction of municipal solid waste. So here in this study we are going to take up for the 2 stages. First one is startup of reactor and then operation of reactor. In the startup of reactor, so there is a problem of wastages conversion into the liquidification stage, because municipal solid waste is a heterogeneous. Huge amount of vegetables and then food waste and then some other wastage are mixed. So the waste is segregated and then it is mixed with cow dung. So in cow dung there is a huge amount of species are available. This some methanogenic bacteria and other bacteria are going to be react with the municipal solid waste, and then municipal solid waste is finally taken into the reactor. So first around 60 to 65 days taken for the startup period. After that the reactor is going to be easily, smoothly producing the biogas. So you are seeing the picture, the lab scale reactor. So in this lab scale reactor, here the bottom portion is the tank, anaerobic digestion tank, and then top portion, so there is a venturi type of that funnel is there. The funnel is going to be convert the biogas, and then untreated wastewater is taken and it is taken for the further treatment. Here we are getting that, but the pH is neutralized during the study, and then biogas is taken from the top side. Here you are seeing the flowchart like that. So here liquefied organic fraction of municipal solid waste is taken into that vessel. That vessel is taken into that bottom portion of the UASP reactor. Finally, it is going to be measured. And measured, there is a gas collection drum. It is going to be measured the huge how much amount of gases. Here, various components of organic fraction of municipal solid waste is shown in the figure. It is a cooked rice, fresh cabbage, tomato, cooked vegetable, cereals and various kinds of fruits and chapati and some other different kind of food stuff. So the percentages also taken here. Okay, that what are the various chemical parameters should be maintained also shown in the slide. And here various parameters, what are the inlet parameters taken for the study, what are the outlet parameters are taken for the study. Finally methane content also, it is shown in the last column. So it is the graphs are given for the biogas production. The biogas production initial stage, it is somewhat less, and pH also maintained in the neutral manner. Here the COD removal rate are around 60 to 80%, and then alkalinity and volatile fatty acids also should be maintained. Finally, we are that loading rate, it is important parameter in the study. If we are loading smoothly, then only biogas production will be smooth. Otherwise, it is going to be disturbed. The biogas production will be disturbed. So here, in this reactor is one of the optimized reactor because we are getting the smooth production without any disturbance of the reactor. Here, we are. I think that the students are going to be use this technique for their study. Thank you.